Welcome to Learn Biology, and today we're discussing RNA synthesis, meaning how ribonucleic acid is created. We have already discussed RNA in other videos, so if you need the structural composition, please check out the other videos. But today we're dealing with exactly how RNA is made. So we'll begin with the first main properties of RNA, which include that RNA is single-stranded. It contains the nitrogenous base, a uracil, instead of thymine, and makes proteins. So the thing we need to understand is that it is single-stranded. This is very important, and the function of it is to make proteins. RNA is used to make proteins. That's why it's very important. And when we say that it contains uracil, so it is taking the place of thymine. So uracil takes place of thymine. So now we have adenine connecting to uracil, and then the standard guanine connecting to cytosine. But that's what RNA is important. And before we go on, we have to understand another thing. The process by which RNA is made is called transcription. So transcription. And what you need to understand is that DNA makes RNA. And why is this? Well, DNA contains all the genetic code. It contains all the genes. But these genes cannot be expressed without the creation of RNA, because RNA creates proteins. So DNA makes RNA. That's what we need to understand, and that is called transcription. So transcription, keep that in mind all the time, transcription. And we're always going to be dealing with only one enzyme. DNA had 4 or 5 enzymes. RNA has only one. It is called RNA polymerase, and we will see its functions in the next slide. So the first step is that, remember, DNA makes RNA. So what happens is that the double helix of DNA unzips. So DNA unzipped, and it is unzipped by the enzyme RNA polymerase. That is what unzips DNA. Now, okay, so here we can see that DNA has been unzipped. This section right here has been unzipped by RNA polymerase. So RNA polymerase. So a few definitions we need to understand. RNA is going to be created by one strand. The strand that RNA uses is going to be called the template strand. It is called the template strand or the sense strand. So the sense or the template strand is the strand by which the RNA is created. In this case, this is the template strand, sense strand. We also need to understand that RNA begins creating its chain. RNA polymerase brings forth the nitrogenous bases to create the RNA. Now, this is a lot to keep in mind, but here we'll label this. So this is the template strand, okay, right here. It has been opened up. Now RNA polymerase, you see this blue, has come in and starts to read the strand. It reads the template strand. By that, there is a section of DNA. Let's say we'll begin right here. This right here says RNA. It is like a flag. RNA begin creation. So we're going to start the RNA right here, and that is called the promoter. So the promoter is a piece of code. The promoter is a piece of code that begins the RNA chain. So it is a section, a code, which begins the creation of RNA. So right here is the promoter, and this is right there. Then we have another code which ends or terminates the RNA chain. So once the RNA chain has been created, there must be a signal that says stop creating the RNA, and that is another chain or another code known as the terminator, because it terminates the creation of the RNA strand. So that's how it works. Now, after this RNA has been created, it must be refined. So the second step of RNA synthesis is known as refinement, which means, what does refinement mean? Well, refinement means taking out or making better and more efficient. So refinement is a process by which the RNA chain is going to become much better. So all the excess code is going to be removed, and security features will be added, and so on. So the first security feature we're going to add is known as the protein cap. The protein cap goes at the beginning of the RNA strand that has been created. So this orange right here is the RNA strand. This cap right here, the protein cap, is going to allow the RNA strand to connect to the ribosome later on. Connect to ribosome. And this is something we must understand here. At the beginning, RNA begins in the nucleus. Then, at the end, RNA is hooked onto a ribosome, because ribosome is the site of protein synthesis. So this protein cap will allow the RNA strand to connect to the ribosome. At the end, we have an adenine tail. This tail right here, so the adenine tail is a security feature that just keeps extra adenines in just to prevent enzymes from eating up the strand. There are numerous enzymes throughout the cell which can damage the RNA strand. So this prevents damage. So prevents damage. And this is known as the adenine tail. It is made out of the nitrogenous bases adenine, series of adenine. Now we must understand, we said refinement is making better and more efficient. For that, the excess code is removed. So introns are known as the excess code. The introns are known as the junk code that have no not useful purpose in the RNA strand. So introns are removed, and exons are very important. These are known as the essential DNA. So the essential DNA, just scratch that out right there. Exons are known as essential DNA, and only exons are kept. Introns are spliced away, and that is what refinement does. Now, once refinement is done, we are finally done. The RNA strand leaves the nucleus and goes to the ribosome, and it attaches to the ribosome, and we're complete with RNA synthesis.